So we're talking about identifying organic compounds for our lab prep for tomorrow. Now again, you guys are going to be solving this mystery for the lab, so you need to be able to know which organic compounds are going to be in which of the samples that we are testing. And again, the organic compounds that we are talking about are proteins, lipids, carbohydrates, and nucleic acids. Now all of these are very important. We talked about their functions and structures in our other video, which I will link here if you need to review, but I wanted to go through just the indicator tests very quickly. So each of these has a different type of indicator test that we're going to be using. Some of them are a little easier, some of them are a little bit more difficult, and some of them, depending on which type of lab setting you have, you may or may not be able to do. Let's go through them one by one. For our protein test, we have what's called Biuret reagent. Now this is a specific type of chemical that's going to test for peptide bonds. And remember, our proteins are made up of bunches of amino acids linked together with peptide bonds. So that's what the Biuret reagent tests for. If it's negative, it's going to be this blue color. It's pretty blue. If it's positive, it'll turn purple, and that will show us if we have protein in our sample. Now, the actual concentration of protein is directly related to the darkness of the color. So if we have a lot of protein, it'll be a very dark purple. If it's a little bit of protein, it'll be a light purple. So next one is lipids. This is our very technical, very important brown paper bag test. Now, I'm sure you've eaten some food before and had it in a brown paper bag or gotten a bag from fast food and noticed that the oils or the fats have sort of stained or gone through the paper. And what it does is it makes the paper a little bit translucent or a tiny bit transparent. And that is our test for lipids that we're going to use. It's not very fancy, but it does work. So you can see here from other science teachers that have posted on the internet, this is what it looks like. If you have butter, which again is a lipid, you'll be able to see through it. And you have to wait a few minutes for it to dry. Obviously, if you spill a little water on any type of paper, it'll make it a little bit see-through, and you can see the water sample there. But a real test for lipids will show some translucence through the bag. You'll be able to see through it a tiny bit. Now we're going to talk about our carb test. Now there's lots of different ways to test for different types of carbohydrates, but first I want to look at our monosaccharides. There is a special solution called Benedict's solution. When you heat it up, will change from blue to a yellowish or orange if it has monosaccharides. Now remember, our monosaccharides are our simple sugars, our sugars that are not going to be in long chains like glucose. So if glucose is present, we would see this change in color. If we go to another type of test, we can also test for glucose with our glucose test strips. Now sometimes we'll see these with just the glucose on them. Typically you might see them in a urinalysis test strip if you're testing someone's urine for lots of different factors. Our glucose test would be at the very top here, and if it changes color anywhere from this light blue to any one of these values. And this is neat because it shows us the different increments, the different amounts of glucose that it is showing in this indicator. You'll be able to say, yes, it is positive for glucose, or no, it does not have any glucose in it. Finally, another type of carb is our polysaccharide, many molecule attached sugar. So this is our sugar with lots and lots and lots of little glucoses connected to each other, or a starch. An easy way to test for this is our iodine test. Remember, iodine is going to be like a brownish or deep yellowish, and if there is starch there, it will turn to a dark purple or a black color. So those are our indicator tests. They're not very complicated, and hopefully you will be able to use these successfully when you come into lab tomorrow. Okay, see you guys later.